We get these new traits from a variety of sources. And some of those sources could be wild germplasm. Germplasm is just a sciencey word for genetic material. So, what looks like weeds in many cases. A lot of the tomatoes and lettuce and strawberries and other things, if you compare it to what we grow and expect to see in a farmer's field or in a supermarket shelf, the wild germplasm looks like a weed. And they're potentially horrible tasting, horrible yielding, don't grow the way we want them. They don't have much of anything that we would recognize right off the bat. But what do they have? They have hidden genes that uh, can produce all kinds of different uh, valuable traits. Disease resistance is often one of the easiest things to find because all you have to do is take samples of wild relatives of your lettuce, your tomatoes, your potatoes, your corn, whatever, and grow them up and spray them with your pathogenic material, your bacteria or fungi. And after you spray them, see which ones survive. And go, okay, that one, that weed, that weedy wild germplasm appears to have genetic resistance to this pathogen. I'm going to try breeding with it and see what happens. But there can be all kinds of other things. Just think about wolves and domesticated dogs. You start with a wolf, and what would you expect? More wolves. What did we end up with? A very short time in terms of genetic history and evolutionary history. Humans have been breeding dogs for only a few thousand years, and some of the breeds very recently within the last 100 or 200 years. And we go from wolves to Great Danes and Chihuahuas and Boxers and pinchers and on and on and on. All of that variety, all of that genetic variation is hidden inside of wolves, and you can breed for it. And the exact same thing is true when you look at these wild germplasms or wild species of all kinds of plants. And you can manipulate them. Things like the brassicas are one of the easy to recognize examples because the same species of brassica includes cabbage, Brussels sprouts, cauliflower, broccoli, Romanesco, and more. It's all just hidden genes in this one species of brassica. And breeders have taken advantage of that. So wild germplasm gives lots of flexibility. It's just a lot of work. You don't know what you're going to get. It's just throwing the genes in the air and go, wow, I wonder what's going to come out of this. And sometimes some really boring or unuseful things come out. And other times, every once in a while, you go, wow, that's kind of cool. That gene could be useful. Land races and heritage varieties. Now, these are varieties that are collected from different indigenous peoples and cultures and countries, collections that are all over the world. So you go to places where people are just growing things and have been for generations. And there are people, even in places where you don't think about heritage varieties, there are people in the Midwest of the United States who still hold on to some of their traditional varieties and still grow their traditional corn and don't grow everything with brand new Pioneer hybrid brand seeds or Monsanto seeds or something like that. They exist. These heritage varieties exist all over the world. And there was a big push, I would say, immediately after World War II was kind of a new age push. The modern day um, genetic explorer. Back in the 1600s, 1700s, there were explorers that would follow around the the royal navies of the world, and they were called naturalists, and they'd be drawing these beautiful pictures of everything they found, discovering species of plants and animals, taking collections and uh, mounting them, pressing them, drying them, doing whatever they could, saving seeds, whatever they could do to learn what was out there. And so kind of died down after a period of time and into the background. It still was going on, but it died down in the background. And then with the Industrial Revolution, there was a big push to understand the genetics of our our food supply. What do we eat? What do we grow? Our food supply is based on a handful of things, from corn and wheat and soy and cassava and um, our fruits and vegetables that are more minor crops, but they're important, especially regionally. And people started to think, we got to do something about this. And there were some efforts in the early part of the Industrial Revolution to collect these land races, heritage varieties, and find wild germplasm. But after World War II, it exploded in the 40s and 50s and even into the 60s, where specialists for damn near everything you can imagine, tomatoes and papayas and cabbage and you name it, they were going to special places in different parts of the world collecting these germplasm samples. The elite germplasm, that's the best of the best that we grow. That's the cutting edge, state of the art, modern varieties. And most of that is proprietary. Companies own it. And so you don't go get a land race from some indigenous peoples in the mountains of Peru. We're talking about stuff that's owned by major corporations primarily. Uh, the elite varieties are the ones that are actually being sold. To give it the name variety, those are the things that are being sold, as opposed to the germplasm, which is the raw materials for making the things that are being sold. And all around the world, there are also seed collections. Where do all these things go? And it's not just seed, it's also clonal materials. And the US government has some stuff, the universities have stuff. I know UC Davis has the world's largest tomato germplasm collection. They are the International Center for Tomato Germplasm, and there's an entire department there. I actually did a lot of work with them and worked for them in some ways for a while. There's also private, nonprofit, or for profit organizations and some seed companies. Some of them, it's more of a hobby, and some of them, it's a passion. So there are some things like Johnny's Select Seeds, or I can't remember the name of them off the top of my head, but there's a number of these that sell varieties to the average home gardener to get something interesting, heritage, unique. So you're not growing the same old everyday tomatoes that everybody else is in your garden. You're growing something historically interesting and different, and um, sometimes different colors and shapes and things that you don't even think of when you think of tomatoes or lettuce or whatever. And behind the scenes, even if they might have a face of, we're all about the home gardener, well, that's their cash flow and it's important. They might have a passion for saving these germplasm samples. So, next we get into the breeding methodologies, and don't get bogged down into the details of this. I just want to talk about some of the more common concepts, and don't worry that I think this is Dutch. Doesn't matter. Yeah, I'm pretty sure this is Dutch, but the picture works. So I have parent one and parent two. Doesn't matter which is the male and female in this picture anyway. And it creates the F1, or the first filial, 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 the first filial generation. And you cross two plants, two parents that you think will give you a good combination of genetic material, and you kind of shake and stir, so to speak, the genetic material between them. How do you shake and stir? You first make a cross, and it's half and half, mom and dad. But then you take that first filial generation and you self it. Either you force self, there's different ways you have to do it with different crops, but let's just assume it's self compatible and you let it self, or you force it to self. And then you get the F2 generation. And then out of the F2 generation, you just grab a number of individual seeds from each of the selected lines that you chose. Don't worry about how you choose those lines, but you just grab some individuals and then you self it again. And then you grab one seed from those individuals. Each individual gets one seed to go to the next generation. In reality, I always did two in case one of them died, got a disease, I had a backup. But the idea is it's one seed, single seed descent. And during these selfing events, basically what you're doing is taking what was half mom and half dad, and you're mixing it up, you're stirring. As they self, they're crossing internally the genetics of the chromosomes, the constituent genes of each of the chromosomes are being shared. And it's stirring what came from mom and dad. And at some point along this line, you decide to make selections. And you could start some of the selections very early, but then you have to make the real selections. So this, you're talking about these bars, you're talking about multiple plants. I'm going out into the field, and I am growing up some plants in groups. So now I'm allowing the generations to grow in several plants that I'm getting from this generation, and I'm making a choice. In this case, everything yellow in the F5 generation I throw out because it didn't have something I wanted, I was looking for. And the black ones are groups of plants that did look good. So I send that to the next generation. And again, the ones with the crosshatch look good, I let them go to the next generation until I finally have one winner from that cross. And that's pretty optimistic. A lot of times, most of the crosses you end up where you throw it all out. You're lucky if out of every 100 crosses you get to one that was the winner. But it's a numbers game. It's a numbers game. But it's single seed descent. Why do I use single seed descent? You are combining the traits of two adapted varieties because at the end, on average, you have half mom and half dad still. That's the key. You're just mixing up mom and dad. And if one of them is a weedy, ugly species that had disease resistance, but it was a wild weed, this would be horrible because what's going to happen at the end? On average, you'd get plus or minus 50% wild weed, and it's going to look ugly. So both mom and dad have to be adapted varieties. They have to look like the finished product you're after, at least close to it. Mom and dad have to be good varieties on their own. They just aren't having everything you want. Mom has a really good variety, but missing something. Dad has a really good variety, but it's missing something. And dad has something that mom's missing. All right, cross them. And see if you can get both of them with those traits in a single plant down the road. Single seed descent, two adapted varieties. And if I had to tell you what to study for the test, that last statement is really the key. Single seed descent, two adapted varieties as parents in order to combine the traits of those two adapted varieties. The next one, bulk selection. This one is you're doing population breeding. And you, in some types of bulk selection, at least the variation that I've used, it's been for a population of things where you want some residual uh, differences. You want some genetic heterozygosity or some genetic variation left when you're done. So, for example, one project I worked on was alfalfa, and alfalfa for grazing livestock. And so, if I'm making a field with alfalfa for grazing my livestock, I don't want everything to be absolutely uniform. I want some of them to grow up a little bit, some of them maybe to grow down a little bit. I want some disease resistance in this one, but a different disease package in this one. So if we do get disease coming through, there's some variation in my plants that will adapt in the natural environment of my pasture. And so what do? I take parent one and parent two, which have all these traits. I get the F1 and I just bulk it. This represents, I just throw all of the children of the F1 into one giant bulk and let them do their thing. Let all the genes cross between plants, selfing, whatever they do. And I do it again. And after a few generations of letting the genes get stirred up, get mixed up, then I start doing some selection of individual plants later. So the bottom line with this, bulk selection is a population type of breeding. And then we have backcross breeding. And before I get to backcross breeding, I think I'm going to take another 4 millisecond break.